Okay, welcome back. Today we're going to tackle DNA sequencing. So this is sort of our first real exploration into biotechnology. This is some stuff that's been around for a decent amount of time, but is still very, very useful and used in every lab ever. I don't know any lab that doesn't use this stuff. So it'll be fun to talk about and kind of get you acquainted with this technology. So just as a review, let's talk about DNA replication. We just talked about it, but just to make sure that we're kind of all on the same page here. So DNA strands separate to go through DNA replication. There's a template strand, which is like the original strand that is part of the original piece of DNA. A primer will start the replication. DNA polymerase that adds the new nucleotides, and then a new strand is formed from that template strand. Okay, DNA is formed in a 5' to 3' direction always, and the whole thing is semi-conservative. Okay, so when we're sequencing DNA, and what that means is we are figuring out the order in which the nucleotides are set up, right? So does it go A, T, C, G, A, T, C, G, or is it a different order? We don't know. That's why we use DNA sequencing to figure out. Our main two pieces of technology that we use in this is polymerase chain reaction, or PCR. Like everybody calls it PCR, but now you know what it stands for. And gel electrophoresis. So PCR is a process where scientists just make hundreds of copies of DNA. Basically, it just forces DNA to go through the replication process, and you just make so many copies of it, so you have a ton of the same DNA to work with. It makes it easier to see, because DNA is so tiny. So when you make a ton of it and it's all saying the same thing, it just kind of adds to your results. And the way that DNA sequencing works is it basically goes through a really similar process to PCR. And then gel electrophoresis is how you separate that DNA by size. So what happens is you put the DNA into this gel, and it is quite literally a gel. It feels like a thick Jell-O, right? Like you can put your fingers straight through it, but it's like really bendy. And then you hook it up to an electrical power source. And as you push that electrical current through the gel, it's going to push the DNA fragments through the gel as well. And they're going to separate based on their size. The bigger the DNA molecule, the heavier it is. So it's harder to move as far. So the longer molecules stay kind of near the beginning, whereas the shorter molecules will get pushed all the way through the gel. And if you're not careful and paying attention, you can push the DNA right out of the gel. So you really have to pay attention when you're doing this, just to make sure that you actually keep all the DNA that you're looking for. And I do speak from personal experience there. Now, what we're going to focus on in this sequencing piece is the Sanger method. This is a sequencing method developed by Fred Sanger, who is pictured there, shocker, white male, pretty recently, right? 1977, that is like not that long ago. And this is kind of the basic and most used method for sequencing DNA. And so it makes this sort of automated cycle sequence, which is really cool. And you might see this in like websites or in other things that we're going to be doing, but it's also called the dideoxy chain termination method. And that is like really important. So deoxy is the type of sugar in DNA. And so it breaks the relationship or the connection that's usually created between the sugars in DNA. So it breaks that and it doesn't allow that to form, and that's like the basis of this method. So just to give a little bit more information about this before we actually view what it is basically you have a bunch of like relatively short DNA fragments that you need to get sequenced. So modified nucleotides called dideoxyribonucleotides, right? So slightly different nucleotides than what are normal nucleotides attached to the DNA strands. And there are 4 types of these DDNTPs, right? You have one that corresponds to adenine, to thymine, to guanine, to cytosine, right? To A, T, C, and G. So you have 4 modified nucleotides that match up with the regular nucleotides you would find in DNA. And you tag each of those modified nucleotides with a fluorescent label. That way, when you're done with all of this, you can very quickly see where the A's are, where the T's are, because they're fluorescently labeled. So you can just look and be like, oh, there's a pink one, that must be A, or whatever color you assign it, right? When those modified nucleotides attach to the DNA fragment, it prevents any new nucleotides from adding on. So it terminates replication at that point, and then you can start to read the sequence. It will make more sense when you see an image of this. So before we move on, I just want to point out that on the top here, this is a normal nucleotide. This is like a very general nucleotide. You see that it has an OH group on the 3rd carbon of its sugar. The modified nucleotide does not have an OH group, and because of that, you cannot go through the reaction that connects nucleotides to each other, and so basically it's like a dead end. Once that connects to the one before it, no new ones can connect to it, and it terminates all replication. So, kind of what I just said, since the 3' carbon doesn't have an OH group, it can't bond to another nucleotide. So normally, you would be able to bond this right here, right, to the next nucleotide. But without that OH group, you can't. So it just totally terminates. It ends, like, stops dead in its track, stops cold. You can't do anymore. So the Sanger method uses these defected nucleotides to make DNA strands of different lengths. By tagging and labeling them, you can identify the base that prevented replication from continuing on. And then you just order the sequences by size, and you get the sequence of DNA that you're looking at. It's like, oh my God, it's so easy, and it's so cool, and like, obviously I'm a fan. So this is what you do. In a little like pipette, you would add in all of your normal DNA, right? This is your normal nucleotides, A, C's, T's, and G's, normal. And then you would add in your modified nucleotides, fluorescently labeled, right? All these fun, funky colors. And then you make a bunch of copies of that DNA template strand. And then when you add in those modified nucleotides, that replication process stops. But because you're making so many copies, the replication is going to stop at different times because the modified nucleotides are connecting to the template strand at different areas. So you have tons of new DNA strands being created, and they're all of different lengths because the modified nucleotides are attaching at different places. So once you finish all of that and all of the modified nucleotides have attached to that DNA, you just put them in order using the gel electrophoresis. You put them in order from, big to small, and you just read it, and that's it. So another image for this, so right, you have the strand that you want to be sequenced. You prepare those 4 reaction mixtures that have the modified nucleotides in there, and then you have all these different lengths of DNA produced. You put it through the gel electrophoresis, and you can see that it's going to stop at different times, which will allow you to read what the sequence was. So what happens is you get, again, this is just another way to show you the Sanger method, right? We want to sequence this strand. As it goes through, you have, this really tiny one was formed, right? The modified nucleotide stopped it right here, and then it will continue on. Modified nucleotide stopped all these at replication at this point, and then you read it, you put it through the gel electrophoresis, right? The smallest fragment is the furthest one down because it's the smallest, so it's going to travel the farthest. And then you can sequence it, right? The pink ones correspond to T, so T, T, G, T, right? You just correspond the letters to the colors. It's as simple as that. We'll practice this a little bit in class, but really I just need you to be able to read a gel from bottom to top, and then that will tell me that you understand how the Sanger method works. So, nice job. We'll keep learning about this in the upcoming classes.